Okay, so considering that some of you might be new to this particular session that being conducted by me, so let me briefly introduce myself. I'm Arveen. I'm a PhD scholar at IIT Kanpur, and I am also somebody who is working in the field of neuroscience actually. So which is why this course really enticed me, and I requested Professor Varadhan to let me be the TA for this course. So in these sessions, what we'll be looking at is previous year questions pertaining to the week for which the session is dedicated. Over here, we'll be looking at week 3 previous year questions. What we'll do while going through these questions is that we'll also understand the concept behind it, so that it will help strengthen that concept in your mind, as well as probably helping you solve the current year questions also. Right. Along with that, what we'll also do is we'll go through certain neuroscience related topics that were touched upon in Sir's lectures, but if you are new to, or if you are very new to this course neuroscience, you might not have a prior knowledge about it. So we'll try to elaborate those topics a little bit. That will be the second part of this session, right? So we'll begin with the previous year questions for now. Um, okay, just a moment. Yeah. So, for instance, the first question pertaining to week 3 lectures was, what scientific advancement in the 1950s actually revolutionized the study of cell biology, right? Was it the invention of light microscopy, or the introduction of electron microscope, or the discovery of DNA structures, or the use of fluorescent staining? Right? So, which one of these was it actually that revolutionized specifically cell biology around 1950s? So, the answer
See,
has to be electron. No, it was not actually the discovery of DNA structure. So, DNA structure, it did help us on a molecular level to understand how information is getting transmitted. But cell biology was highly benefited by the introduction of electron microscope actually. Why? Because it was the first time when this electron microscope was used on animal or plant tissues. It was the first time that we could actually visualize the intracellular structures such as mitochondria, ER, the ribosomes, the nucleus, right? It was the first time actually that that could happen. Even if we were able to stain these structures before this time and observe these tissues or cells under light microscope, the resolution could not be as much that it would actually aid us in viewing these separate structures, right? So it was the first time that we could actually visualize them, which is why this is the advancement that is being talked of. But yes, if we talk about molecular biology, then C would be the correct answer. That is discovery of DNA structure, right? Or if we talk about genetics also, right? So then this would be a better answer, C, right? So invention of light microscopy is out of picture because this had been around for a while during that time, right? Uh, Even by 1950s, the electron microscope had already been around for around 2 decades, right? So this was the man who was accredited with the discovery of, or invention rather, of electron microscope, right? His name was Ernst Ruska. He was actually awarded the Nobel Prize in Physics, right? He was a physicist in Germany, right? So he he was not designing this particular microscope to look at tissues or cells per se, but it really aided in the biologists at that time to utilize this equipment for this purpose also, right? So, born in 1906, right? He, at the time that this award was actually given, he was affiliated to Max Planck in Berlin, Germany. And the motivation of for his prize is as it is written over here for his fundamental work in electron optics and for the design of the first electron microscope, right? So he was a physicist who actually designed it. Now, uh, how did he come upon doing this? Let us understand a little bit about that also, right? So, why he wanted to design it was because earlier, very small objects could be studied in microscope, but when the objects become as small as the wavelength of light itself, an image can no longer be produced, right? Because what we are seeing is a reflection of these light rays, right? So if the wavelength of the light itself is more than the structure that we are trying to identify, then the image is no longer visible, right? But what he discovered was that the beams of electrons behave as waves with wavelengths shorter than visible light, opened up new opportunities. So Ernst Ruska discovered that a magnetic coil could be used as a lens for electron beams and developed the first electron microscope in 1933, right? Um, Almost 2 decades before it was actually used in biology. It captures images of extremely small objects by means of electron beams that are directed towards an object and captured on a screen, right? So, that was his motivation of doing this, right? He wanted to look at different kinds of particles. He might even have thought that maybe he will be able to visualize, atoms and nuclei, the chemistry and the physics part of it. So even that he might be able to visualize, right? Maybe that was the objective with which he actually created it, but it has really aided our study and our discoveries as well. Okay, so the second question was who introduced the term synapse in 1897, right? So was it Santiago Ramon y Cajal or Charles Sherrington or René Cottier or George Coyle, right? So, in one of his books or manuscripts, I don't recall exactly. Charles Sherrington was the one who actually introduced the term synapse, right? So he used this term synapse to basically define the region which is responsible for passing information from one neuron to the other neuron. Now we know that this neuron which passes information is a presynaptic neuron, right? It has certain neurotransmitters. Most often it has certain neurotransmitters that it releases at the end of the axon, which are basically recognized by this postsynaptic neuron, which is basically the neuron that is receiving the information. So the dendrites of this postsynaptic neuron have receptors for these neurotransmitters that are released by the presynaptic neuron, and that is how the message is transferred from one neuron to the other, right? And this region where this neurotransmitter is released into and bound to, this is known as synaptic cleft. Okay. And so this is basically a synapse, the region that is helping in transfer of information from one neuron to the other. Then the third one was what key discovery about the chemical synapse was reported in 1954, right? So was it the structure of myelin sheath or the visualization of synaptic vesicles and their role in neurotransmitter release or the identification of ion channels in the neurons itself or mechanism of action potential propagation, right? So over here the interesting thing is that even if you don't remember discoveries by the year that it was done in, you can still rule out a lot of options, right? So that the most sensible option then you can choose as the answer you want to give. So over here, for instance, the structure of myelin sheath has nothing to do with chemical synapse being present or not, right? So if you are clear with that concept, that myelin is essentially for an insulation that is present around the neuron. It helps in conduction of the electric potential, but doesn't have a role in chemical synapse per se, right? So you can rule out option A, right? Similar is, for instance, is action potential propagation, um, which synapse helps in, but it helps it via neurotransmitter, right? So direct action potential propagation doesn't happen. And similar was identification of channels in neurons. So these ion channels are present throughout the neuron, right? Throughout the axon, throughout the dendrite cell body. So it is not particularly at the synapse that it will help, right? But the visualization of synaptic vesicles, which was possible due to the EM microscopy that was done at this era, helped reaffirm that yes, chemical synapses were present, and these synaptic vesicles will probably be, holding neurotransmitters within them that are released into the synaptic cleft, right? So over here, for instance, you can see this is an example of a neuron which is going and innervating a muscle fiber, right? Over here, in a zoomed in version, you can see that there are innervating regions wherein this axon, there are these different ends of this axon that are present, right? They are going and they are basically, coming in contact with something that is present on the muscle itself, right? Over here, this is how it has been represented, that these axons, they have an end bulb region that which is a little, uh, bulged. It is a little bulged region at their ends. This is the region where a lot of these vesicles are present. These vesicles contain neurotransmitters, right? These are synaptic vesicles. In this particular case, it is containing the neurotransmitter ACH, that is acetylcholine, right? And these vesicles actually fuse with this side of the membrane and release acetylcholine outside, which is then recognized and bound by these acetylcholine receptors, which are present on the actual muscle fiber, right? So that is how the conduction actually happens. So these acetylcholine and acetylcholine receptors that we were seeing earlier, this is a basically electron microscope representation of it, right? So you can see over here that there are these, let me just zoom it even further. I was, yeah. So if you can see this, these rounded structures inside, these are the vesicles, right? So there are these n number of synaptic vesicles that are present inside, right? This is the end bulb of this axon. These black colored worm-like things that you can see outside is the staining, which is recognizing basically the acetylcholine receptors. So these are the receptors that you can see, and these are present on the muscle fiber, right? These regions of the muscle fibers, as we were seeing earlier. So uh, this is what an EM representation looks like for this image that we were seeing earlier, right? So acetylcholine receptors in the vertebrate neuromuscular junction are concentrated at the top one-third of the junctional folds, right? So you can see the fold is like this, right? But receptors are generally on the top itself, right? There are very less receptors that are on this, like on this part of the fold, right? They are generally on the top of the fold. This receptor-rich region is characterized by an increased density of post-junctional membrane. The autoradiograph shown here was hair was made by first incubating the membrane with radio-labeled alpha bungarotoxin. So it is basically a poison from a particular snake which binds to the acetylcholine receptor. So it is used to stain this receptor. Radioactive decay results in the emittance of a particle that causes overlaid silver grains to become fixed along its trajectory, which are these black grains, right? Okay, moving forward. So, what organism did Hodgkin and Huxley use to study action potentials? Was it the frog or rat or squid or fruit fly? So, it is pretty simple over here, right? The answer to this is squid. It was something that they had picked up from another scientist who had pointed them into the direction of squid, wherein they had realized that the squid actually has a squid giant axon that we now call it, which is an axon whose diameter is 1 mm, right? So 1 mm is measurable even through sight, right? Like you should have a very fine scale for it, of course, but it is even visible up to some extent, right? Unlike all other axon or all other neurons that they had been trying to study earlier, right? So it was a good model organism for this. So this is a representation of the concentrations of specific ions in the squid giant axon, right? So their relative concentration in the cytoplasm that is within the uh, neuron and outside in the extracellular fluid. And as we already know from our previous discussion that potassium is much more present in it is much more surplus within the cytoplasm of the neuron as compared to sodium and calcium and a few other ions which are more in concentration in the extracellular fluid than within the cytoplasm, right? And these are the equilibrium potentials of each of these ions. Okay, and important thing to notice that the actual equilibrium potential, that is resting membrane potential, is closer to this potassium ion, right? So it is actually minus 70 millivolt in case of the squid, which is much more closer to the potassium ion than any other. So there are multiple reasons for that. There is a proper mechanistic concept which it is related to. Um, Probably if I have not kept it in today's, I think I have skipped it. So we'll discuss that in the upcoming lecture then, right? Okay. So question number 5 is which neurotransmitter did Rene Cotia focus on in his research at the neuromuscular junction? Right. So lucky for you that we have just seen an example of neuromuscular junction, and you should have already revised which neurotransmitter is more commonly found there. So is it dopamine, acetylcholine, serotonin, or glutamate? Right. So since we have already gone through this, it is actually acetylcholine, right? So acetylcholine is the more common neurotransmitter, especially found at the neuromuscular junctions, right? So this is the one that he focused on. Yeah. So the next question is, uh, hold on. Yeah. What principle explains the rapid change in ion flow during an action potential according to Hodgkin and Huxley? So, was it the synaptic vesicle theory or the ion flow model for sodium and potassium or sliding filament hypothesis or genetic information flow? Right? So, action potential also we have not studied in detail as of now, but I have kept a section that is dedicated to study of action potential and study of this particular model that is being talked about in this question. So, for now, I will discuss the answer, which is basically ion flow model for sodium and potassium, right? So, action potential essentially, as I have already said, we have not discussed it as yet, but it is basically a particular threshold needs to be crossed for an action potential to be attained, right? And the changes that come to drive or to withdraw an action potential generally happen due to the ions that are flowing into and out of the extracellular uh, fluid, right? Over here, the most discussed ones and the most abundant ones actually are sodium and potassium. The third most abundant is chloride. And later, there were many other ions also that came into picture over here. But these are the most abundant ones, right? So Hodgkin and Huxley, when they were speaking of it, they have focused solely on these two ions, right? So, but ion flow model is the accepted model. So the sequential opening of the voltage gated sodium channels. So you can see that first there is a peak for sodium channels, then this peak is going down. Once it is going down is when the potassium channels are opening, right? So probably they are exclusive to one another. At the same time, they can't open, right? So one of Hodgkin and Huxley's great achievements was to dissect the change in the conductance due to an action potential into separate components attributable to the opening of sodium and potassium channels. The shape of the action potential and the underlying conductance changes can be calculated from the properties of the voltage gated sodium and potassium channels. Right, so there is actually another set of experiments that has been done wherein there are some chemicals that have been used to restrict the sodium channels from opening. Then some others that have been used to restrict the potassium channels from opening. How that has helped us is that if only sodium channels are present, then what will happen to the action potential? If only potassium channels are present, then what will happen to the action potential? So we have that very descriptive study also, right? Which also we'll look into later in this session. So, the seventh question is, what did Goldman-Hodgkin-Katz equation formalize, right? Did it formalize the nerve impulse speed, or the resting membrane potential, or the membrane potential differences from the ion permeability and gradients, or the synaptic transmission efficiency, right? So, this equation, again, what it helped was it looked at different ions and their permeability. And at this time, what we knew was that probably, uh, just a moment. Yes. So what came to be known after the work of Goldman, Hodgkin, and Katz was that different ions have different permeabilities, right? And because the concentration is different in the extracellular fluid or the cytoplasmic fluid, they have gradients, right? So, for instance, potassium will have a gradient from inwards to outwards, right? But sodium will have a gradient from outward to inward because outside its density is much more than inside. So there are these differences that exist, which was actually formalized by this equation, GHK equation, right? So what it said was, this was the final proposition. Let us come read this and come to this conclusion then. So Alan Hodgkin. So what it says is Alan Hodgkin and Bernard Katz used the Boltzmann equation to analyze changes in the membrane potential in the squid giant axon. What they did was they measured the variations in the membrane potential in response to systematic changes in the extracellular concentrations of sodium, potassium, and chloride. They found that if this membrane potential was measured shortly after the extracellular concentration is changed, right? Then potassium has a strong effect on the resting potential, while chloride has a moderate effect and sodium has little effect, right? So based on this data, what it okay, this word is fit, right? They basically did this calculation based on the resting membrane potentials and the relative permeabilities of these ions, and they came up with the following permeability ratios, which is the permeability of potassium ion to sodium ion to chloride ion is 1 is to 0.04 is to 0.45, right? Which is what this final statement over here says, right? That sodium, potassium has a strong effect on resting potential, chloride has a moderate effect, while sodium has little effect, right? Thereafter, the next question is which method introduced by Kenneth Cole was critical for studying nerve excitability? Was it voltage clamp technique, or electron microscopy, or fluorescent dye staining, or atomic force microscopy? Right? So, Kenneth Cole, what he actually did was he discovered the voltage clamp technique, and this technique he later taught to Huxley, who basically used it well to understand the nerve excitability, right? So, voltage clamp technique was developed by Kenneth Cole in 1949 to stabilize the membrane potential of the neurons for experimental purposes. It was later used by Alan Hodgkin and Andrew Huxley in early 1950s in a series of experiments that revealed the ionic mechanisms underlying the action potential. Right? So let us understand this technique. And it is not brief, it is a detailed description of this particular technique. Right? So, what is this voltage clamp and why was it required? So, if you remember Sir's lectures, also, you would have recalled that before this, also, we could make out what is the local action potential at a particular region, right? But that was an extracellular recording. So, extracellular recording will not be accurate, 100% accurate it will not be, or it will not be very accurate also, right? So what this particular technique helped us do was that it helped us understand the intracellular potential of a particular neuron, right? That is why it is so famous. So what happened over here was that the voltage clamp basically permits the experimenter to clamp the membrane potential at predetermined levels, preventing changes in membrane current from influencing the membrane potential. By controlling the membrane potential, one can measure the effect of changes in the membrane potential on the membrane's conductance of individual ion species. Right? So what they were actually doing was they were uh, uh, putting this membrane potential on hold, right? So that if first light current is also coming, but the membrane potential is staying put, right? It is not changing very rapidly. So the voltage clamp consists of one intracellular, one extracellular pair of electrodes used to measure the membrane potential, and one intracellular and extracellular electrode used to pass current across the membrane, right? So what the setup looks like is this, right? So there is one intracellular, extracellular electrode within this region which is measuring, and one that is outside which is helping to pass the current, right? This current is being passed by another set of electrodes. So through the use of negative feedback amplifier, you can see that there's a feedback amplifier that is given over here. The voltage clamp is actually able to pass the correct amount of current across the cell membrane to rapidly step the membrane to a constant predetermined potential, right? So whenever it is a physical property and it will keep on changing, right? So whenever, for instance, the potential over here is dropping, the because we are recording what is getting dropped, right? This feedback will go to the amplifier and it will know that okay, this much has dropped, so this much current we need to give to put it back to normal, right? So that is what it helps in doing. So depolarization opens voltage-gated sodium and potassium channels. What is depolarization? Again, we have not studied action potential, but the resting membrane potential generally, especially in this case, is around minus 70 millivolt, right? So when it goes towards zero, right? The potential is changing, right? That is known as depolarization, right? From minus 70, it is going towards zero. That is known as depolarization. So what happens in depolarization? It opens the voltage-gated sodium and potassium channels, which initiate the movement of sodium and potassium across this membrane. This change in membrane current ordinarily would change the membrane potential, but the voltage clamp maintains membrane potential at a predetermined level, right? So that is what this feedback loop is for. When the sodium channels open in response to a moderate depolarizing voltage step, an inward ionic current develops because sodium ions are driven through these channels by the electrochemical driving force. So it is taking these ions inside the cell membrane, right? The Na influx would normally depolarize the membrane by increasing the positive charge on the side of the membrane and reducing positive charge on the outside, right? Because sodium is positively charged, if it is going from outside to the intracellular fluid, then the intracellular fluid will become more positive, right? It is at minus 70, it will start becoming more positive, and outside, because there is a depletion of so many of these positively charged ions, it will start becoming negative, is what is uh, the general understanding. So, but over here, this voltage clamp, because it is letting this voltage stay put, right? It intervenes in this process by simultaneously withdrawing positive charges from the cell and depositing them in the external solution, right? So, what it does is, because positive charge is increasing inside, it will throw some of these positive charges outside also into the external solution, so that that change that we discussed about is not happening anymore. By generating a current that is equal and opposite to the ionic current through the membrane, the voltage clamp circuit automatically prevents the ionic current from changing the membrane potential from the commanded value. As a result, the net amount of charge separated by the membrane does not change, and therefore no significant change in membrane potential can actually occur, right? So the voltage clamp is a negative feedback system. A negative feedback system is one in which the value of the output of the system is fed back as the input to a system and compared to the reference value. So any difference between command signal and output signal activates this feedback loop, which is a controller that automatically reduces the difference. Thus, the actual membrane potential automatically and precisely follows the command potential, right? So that was the voltage clamp and how that works, right? And how it has been used to actually study the neurons. We'll discuss in another session, right? So then comes the question, what was the finding of Bernard Katz's 1939 monograph on nerve excitation, right? So was it the development of myelin, electrical properties of the nerves, neurotransmitter release mechanisms, or chemical synapse structures? Now, this is a tricky one. You will have to remember this in order to answer it, which is the correct answer for which is electrical properties of nerves is what was actually um, mm, revealed by Bernard Cart's 1939 monograph after nerve excitation, right? Then question number 10 is, what technological breakthrough allowed Hodgkin and Huxley to measure action potentials accurately, right? So again, it was partly discussed when we were discussing the voltage clamp. Was it the cathode ray oscilloscope or fluorescent microscopy or Nernst equation calculator or the confocal microscope? So the correct answer is cathode ray oscilloscope. If you uh, remember this place, right? There is an oscilloscope even at play over here. So we'll understand how, right? So how this membrane potential is actually recorded. Now let us look at this. So reliable techniques for recording the electrical potential across cell membranes were actually developed in the late 1940s. These techniques allowed accurate recordings of both the resting membrane potential as well as the action potentials, right? So over here, for instance, you can see two nerve cells. Over here, there is an extracellular electrode which is outside the cell, or an intracellular electrode when it is inserted into the cell, right? So when it is not, so this is meant to be an intracellular electrode only. So when it is not inserted into the cell, the oscilloscope over here and voltage amplifier looks something like this. But once it goes into the cell, then there is some reading that this voltage amplifier and the oscilloscope actually gives, right? So this is the resting membrane potential that is probably present, right? So it is a negative also. The glass micropipettes filled with concentrated salt solution serve as electrodes and are placed on either side of the cell membrane, like this, right? So wires inserted into the back ends of the pipettes are connected via an amplifier to an oscilloscope, which displays the amplitude of the membrane potential in volts, right? So that is a function of oscilloscope, that it is giving an output of the reading of the membrane potential in volts, right? So because the diameter of the microelectrode tip is very small, it is less than 1 micron, it can be inserted into a cell with relatively little damage to the cell itself. So, when both the electrodes are outside the cell, there is no electrical potential differences recorded. As you can see over here, it is at zero, right? But as soon as one microelectrode is inserted into the cell, oscilloscope shows a steady voltage, the resting membrane potential. In most nerve cells, that resting membrane potential is approximately minus 65 millivolt over here, right? So, this is what the display of this oscilloscope looks like, right? So earlier it is at zero, and as soon as it is inserted, it is showing at around minus 65. So the membrane potential can be experimentally changed using a current generator connected to a second pair of electrodes on one intracellular and the other one extracellular. While the intracellular, when the intracellular electrode is made positive with respect to the extracellular one, the pulse of positive current from the current generator causes positive charge to flow into the neuron from the intracellular electrode, right? So then what will happen if positive charge is flowing? Then from negative, it will start becoming a little more positive, right? So this current returns to the extracellular electrode by flowing outward across the membrane. As a result, the inside of the membrane becomes more positive while outside of the membrane becomes more negative, right? So if that happens, what will happen? It will move a little towards the positive side like this. This progressive decrease in the normal separation of charge is called depolarization, right? So depolarization is what? Over here in this term, it is towards, moving away from the resting membrane potential, which is moving away from minus 60. So if it is going towards this direction upwards, it is going to be depolarized. So depolarization looks like this. Over here, you can see that if it is a little bit depolarized, right? It will, give something of this sort, which is basically an electrotonic or a graded potential, like we had discussed in Sir's lectures. But only after this potential is able to surpass a particular threshold, will it actually become an action potential, and only this action potential can be uh, propagated across neurons, right? At least through chemical synapses. So small depolarizing current pulses evoke purely electrotonic or passive potentials like this. In the cell, the size of the change in potential is proportional to the size of the current pulses, right? Over here, that is the case, which happens. But a sufficiently large depolarization current triggers the opening of the voltage gated ion channels, right? And the opening of this channel leads to action potential, which differs from electrotonic potentials in a way in which it is generated as well as its magnitude and duration. Right. So, action potential will be of a certain magnitude only. It will not be like if, say, 10 was the threshold of this current to be produce an action potential. Sorry, let me start from the basics. So, say that I was giving five of this current and it was showing this much hill. Then I was giving seven of it, it was showing this much hill. So it is increasing, right? But if I give 10 of this current, right, it is showing this level and it is giving an action potential finally. Even if I give 20 of this, it will still go this much uphill and it will still have the same action potential at that level also. So action potential is not graded, like it will be of the same amplitude, same magnitude, same duration. That is the beauty of it, right? So reversing the direction of the current make the intracellular electrode negative with respect to the extracellular electrode, which makes the membrane potential more negative, right? So this increases the charge separation, which is called hyperpolarization. So it is going more negative in this case. So that is what is hyperpolarization, right? There is no action potential in that direction. The response of the cell to hyperpolarization are usually purely electrotonic. As the size of current impulses increases, the hyperpolarization increases proportionately, right? Hyperpolarization does not trigger an active response in the cell, so no action potential is triggered in this case. So that is the end of the first part of the discussion. It took almost an hour. There is a second part of the discussion, which talks about certain things that have been spoken about in Sir's lectures, but we'll elaborate more on that over here, right? Just give me a short.
Hello, am I audible? I guess this is the break. Last few words she said was not audible. Guess if it is a live discussion, am I audible, guys?
I'm sorry I missed that. I think uh, someone was speaking.
Hi, yeah, my name is Divakar.
Yes, Diwakar.
Yeah, the last few words was missing. Like you said, it was some kind of a break, right? Like we are having.
Oh, yes, yes, yes. Sorry. I just wanted to have water, so I just removed my earphones, which is why I couldn't hear you also. So I was just having water. That was the end of it. I'll just resume with the meeting, right? So let me reshare the screen once. Okay, so I'll go forward with the discussion session also. So, in case you guys have any inputs, any problems that you want to discuss, or that you want me to discuss any particular concept, right, that I have not kept as yet. So, I had made a list of things that I need to include in the session. I couldn't fit all of them uh, due to this time constraint that I also had while preparing the session. But if you want, I can obviously include it in the upcoming session. Um, so, in case there are certain things that you want to be included, particularly you want more clarity on a particular concept, I can always do that. So, you can let me know in that case. Right? So for now, what we'll be looking at is first, because Sir had laid a lot of emphasis on Bernard Katz and how you should read a little more about him. So I have kept a very brief, description of Sir Bernard Katz. And it is something that I have picked up from the Nobel Prize organization's website itself. So, who was he? He was basically a scientist who was awarded with Nobel Prize in physiology or medicine in 1970. Right? What was the motivation of for him being awarded this prize? It was for the discovery concerning the humoral transmitters in the nerve terminals and the mechanism for the storage, release, and inactivation. So, neurotransmitters is what was their major study area, and that is what they had been awarded the Nobel Prize for. Right, so he was awarded the Nobel Prize along with Ulf von Euler and Julius Axelrod. And I have already read the motivation for their prize, right? So, it was something to do with neurotransmitters and them being present. So, the nervous systems of people and animals consist of many nerve cells with long extensions or nerve fibers. The signals are conveyed between cells by small electrical currents and by special substances known as signal substances. The transfer occur via contacts or synapses. So these substances that are being talked about till here are neurotransmitters basically, right? So in 1950s, Bernard Katz studied how impulses in motor neuron activate muscular activity by measuring the variations in the electrical charges. For example, he showed how the signal substance acetylcholine in synapses is released in certain amounts, right? So he was working mainly on this NMJ and acetylcholine as a neurotransmitter and acetylcholine receptors to understand basically how that neurotransmission is happening. Then, on a little unrelated topic, what next I have kept is the Nissl substance. So why I have kept this is because for people who are actually, performing experiments related to neuroscience, you will know that this is one of the basic biochemical stainings that we do, Nissl staining, it to identify neurons, right? And why I thought of keeping this over here is because when we were discussing about the EM structures and how this EM was being done at the neuromuscular junction or in the neurons, CNS neurons also, there was this substance that was recognized, right? A darkly stained substance within the cell body of the neurons that was getting recognized. It was known as a Nissl substance. It was termed as that. So, what was this, right? So, these Nissl bodies are basically discrete granular structures in the neurons that consist of rough endoplasmic reticulum, which is a collection of parallel membrane-bound cisternae studded with ribosomes on the cytosolic surface of these membranes. right? So it was actually named after Franz Nissl, who was a German neuropathologist who invented the staining method bearing his name, which was Nissl staining, right? So what Nissl staining actually looks like is this, right? So these are the Nissl bodies that you can see, this purplish colored hue, right? So these are photomicrographs of Nissl bodies, which are indicated by the arrows over here in the cytoplasm of motor neurons in the anterior horn of the spinal cord. So, when you dissect the spinal cord, you take a small region of the spinal cord. This is, and the region that does have the cell bodies of these neurons is the anterior horn of this spinal cord, right? And there, when you are staining with Nissl staining, what you'll see is these kinds of Nissl substances, which are basically marking the cell bodies of these neurons, right? Thereafter, we come to the discussion of types of synapses, right? So, when we talk about types of anything, it could be in different perspectives or different senses that we do this, right? So, before we talk about synapses per se, let me just tell you, like, type of anything could be in different perspectives, right? How we can determine that is that what are, say, the types of colors, right? So it could be, based on primary colors. One type is primary colors, or this is this, secondary colors, or this is this, tertiary colors, or this, right? Or it could be the classification could be based on types of colors, right? So blue will have like navy blue and sky blue and teal and turquoise, and yellow will have canary and mustard yellow, and all these different kinds of yellows. So it could be different based upon what we are looking at. So similarly, we are going to discuss different types of classifying synapses also. The first type is how it was earlier defined morphologically, right? So when we looked at it, it looked different. There appeared to be two kinds of synapses. And this was basically defined as type 1 and type 2 synapse by Gray, right? So according to this, what was known was, or what was thought was, type 1 is usually excitatory. It is exemplified by glutamatergic synapses, and type 2 is usually inhibitory, which is exemplified by GABAergic synapses. So GABA being a inhibitory neurotransmitter and glutamate being an excitatory neurotransmitter, right? So, for instance, over here, you can see there are type 1 and type 2 synapses over here. So, this is thought to be excitatory, this is thought to be inhibitory over here, right? So, differences included the shapes of the vesicles that were present within it. So, over here, you can see that there are round synaptic vesicles in type 1, there are flattened synaptic vesicles in type 2. The total area of the active zone, the width of the synaptic cleft, all this was different over here, right? The presence of the dense basement membrane, right? So over here, there is a modest basement membrane, but over here, it has a very dense basement membrane, the type 1 one, right? So type 1 synapses typically uh, contact specialized projections on the dendrites called spines, and less commonly contact the shaft of the dendrites, right? So generally they go on spines and not on the shafts of the dendrites. But type 2 synapses often contact the shafts of the dendrite or even the cell body itself, right? As is shown over here. Then there are other kinds of synapses also that are present. These are the functional, like if we functionally define synapses, there will be two kinds of functional synapses. They could either be electrical synapse or chemical synapse. right? So difference between the distance between the pre and postsynaptic cells, cell membranes, in case of electrical synapse is almost negligible. It is 4 nanometers only, right? Because they are actually connected by these gap junctions in between them physically. Sorry. Yeah. But in case of the chemical synapses, this distance is more. It is around 20 to 40 nanometer, right? The cytoplasmic continuity between pre and postsynaptic cells is present in case of electrical synapses, but it is absent in case of chemical synapses. The ultrastructural components that are seen in case of electrical synapse are gap junction channels that are connecting these pre and postsynaptic cell membranes. While in case of chemical synapses, there is a presynaptic vesicles that we can observe the active zones, which is the synaptic cleft and the postsynaptic receptors that we can see, right? The agent of transmission through this or across the synapse, in case of electrical synapse, will be an ion current, or in case of chemical synapse, it will be a chemical transmitter, often a neurotransmitter. The synaptic delay is virtually absent in case of electrical synapse because it is directly passing from one cell to the other without any intermediate agent for the passage. But in case of the chemical synapse, because there is an intermediate agent or the neurotransmitter, that is why there is a significant delay. Usually it is around zero point 0.3 millisecond, or sometimes it is even up to 1 to 5 milliseconds or even longer. And the chemical synapse can only be unidirectional, right? Because it has to go in one direction from where the synaptic vesicles or neurotransmitters are actually present to where they can be taken or received by receptors, right? But in case of electrical transmission, it can be bidirectional also because the current can flow in both these directions. Then we come to the neuromuscular junction. Please give me a moment. Yeah, so I reshare the screen. I'm sorry for the interruption. I'm not feeling too well. Yeah. So then we come to the neuromuscular junction, or the NMJ as it is termed. Right, so we have already got through the schema previously in our PYQs discussion, wherein we can see that there is a neuron or a motor neuron whose cell body actually resides within the spinal cord, right? There is an axon that it projects out, which goes and it innervates the muscle fiber, right? Over here, this is the muscle, this is the end bulb of the swollen end bulb of the axon, and this is the muscle fiber which contains these receptors for the neurotransmitter over here, acetylcholine, right? So that is what the NMJ generally is, right? A mature NMJ. But how is it actually formed, right? So generally, it up like during the developmental stages when this innovations are actually happening, and these receptors are still getting bound and formed. What happens is a growth cone approaches a newly fused myotube, right? So muscle is also just formed right now. So this is a myotube stage, right? So this myotube, after it is formed, there is a growth cone from this neuron whose cell body is within the spinal cord. There's a growth cone that is coming, which is approaching this myotube, right? Then what happens? It forms a morphologically unspecialized but functional contact, right? So it is unspecialized because it is just lying on top of this myotube, but it is functional. It can release neurotransmitter, and this has receptor to receive those neurotransmitter, right? Further, what happens is the nerve terminal accumulates synaptic vesicles. So there are more vesicles that come in. It starts accumulating a lot of these vesicles, and a basal lamina forms in the synaptic cleft. So this basal lamina formation starts occurring, right? As the muscle matures, so this was also a myotube, it was not a mature muscle fiber. So as the muscle matures, the multiple axons converge on a single side. So there are multiple of these axons that are coming and trying to, come to this side and reside over here. Finally, all axons but one are eliminated, surviving a and the surviving terminal matures, right? So what the myotube also does is that it only has an allowance for one axon terminal, right? So it eliminates all these other axon terminals. So the axons that do not at all form any contact with the entire muscle, right? Um, it is signaled to their cell body that no contact has been formed, and they eventually get pruned away during this developmental stage itself, right? And this surviving terminal, axon terminal, it matures into what we have seen as the actual NMJ, right? So this is what it looks like. At the mature neuromuscular junction, as you can see over here, pre and post synaptic membranes are separated by a synaptic cleft that contains basal lamina and extracellular matrix protein. So this is basal lamina, right? There are matrix proteins within this basal lamina itself. The vesicles are clustered over here at presynaptic sites, release sites, and transmitter receptors are clustered in the postsynaptic membrane. And the nerve terminals are coated by Schwann cell processes. So Schwann cell is basically like myelin sheath, right? Mm, uh, no, so it is like oligodendrocyte in the central nervous system. So, like oligodendrocytes make up myelin sheath, Schwann cells in the peripheral nervous system, they make something similar to myelin sheath. Yeah. Then there are also disorders of NMJ, right? So if this NMJ is not proper, then there are certain diseases such as myasthenia gravis that can occur. Um, So this is a case of a normal neuromuscular junction that we saw. But in case of myasthenia gravis, what you can see is that till here, the neuron part is almost similar, right? There are mitochondria, there is a neuron end bulb, there are synaptic vesicles over here, there are fusion regions over here. But the junctional folds in the muscle fibers are not formed or they are not defined, right? So what happens is at the neuromuscular junction, the acetylcholine is released by exocytosis of synaptic vesicles at these active zones in the nerve terminal. This acetylcholine flows across the synaptic cleft to reach the receptors that are concentrated at the peaks of junctional folds. Acetylcholine esterase, which is an enzyme that is present over here in the cleft, rapidly terminates transmission by hydrolyzing acetylcholine. Why this happens is so that the postsynaptic, terminal is not continuously activated, right? It has a time period only in which it is activated and then it stops. But a myasthenic neuromuscular junction has a reduced number of acetylcholine receptors, simplified synaptic folds, and widened synaptic space. So the space is also more in between the the neuron and the muscle. but it has a normal nerve terminal, right? Due to this, the conduction cannot be done properly, and the muscle actually starts to die because it is not getting signal from the brain to contract or relax or anything, right? So it starts to die, and that is known as myasthenia gravis, right? Then the study of membrane potential in neurons, right? So, okay. We'll go through this. As we have already spoken, there are some ions which are more concentrated outside, such as sodium, calcium chloride ions, but there are others, such as potassium and some other anions, which are more concentrated inside, right? Inside the neuron. And this is also something that we have already discussed, that how we are going to measure the membrane potential in these neurons with the help of micro electrodes as well as oscilloscopes, right? And we have already seen that how this electrotonic potential happens, what is hypopolarization or depolarization. So we have just seen that, so I am skipping it. Why we did restart the discussion of this part again was because we want to understand what determines the speed of change of the membrane potential, right? So it could be one direct thing, which is the membrane current itself, right? After that, we want to discuss the time constant and the length constant. Right, so these were two topics that were touched upon in your course, but they were not discussed, right? But for somebody who is new to neuroscience, you might not understand the relevance of the time constant and length constant. Over here, also, we have missed a lot of discussion prior to this circuit setup regarding what particular elements can be added to the circuit, right? Over here, what you can consider is that because this is a plasma membrane, right? This is a receptor in between the plasma membrane. So, since it has a potential difference or a resting membrane potential, because it has one kind of potential outside and one another kind of potential membrane potential towards the inner side, right? It can be thought of as an electrical circuit, right? So if we are talking about an electrical circuit, okay, so these are things that require a separate class itself, right? So Professor Nitin has also taught this in the second unit of his lectures of this course, Neurobiology. So, in case you are interested in studying that, we will try to skip a lot of it and we will try to make sense of how we can understand time constant and length constant, right? Okay, one second. Okay. Yeah. So, if we suppose that a constant current is applied, I probably remove the earphones. I think that will help. Yeah. So, suppose we apply a constant current, right? And this current flows from the inner part of the membrane to the outer part of the membrane, right? So, and if we are considering that this membrane has a resistor and a capacitor, right? So, initially, there will be more current that will flow through the capacitor, right? Because resistor is not the general way that the current will go to, right? But once it is flowing through the capacitor, what will happen is that there will be a difference, or the the voltage will get, current will get accumulated across its across this capacitor, right? So once it starts getting accumulated, the it will also by that time start flowing through the resistor, right? So capacitor, it will develop a voltage. So the current through resistor will start flowing and it will increase. So the current through capacitor will eventually decrease and current through resistor will increase. Eventually, all the current that we are applying will go through the resistor and the capacitor will reach a stable voltage. Right? So you will understand this better if you have understood already that how we are uh, taking analogs of the living cell to this circuit, current circuit, right? So, what we have to suppose over here is, or what we have to think over here is, sorry, that the current source is removed, right? So, if the current source is removed, what will here happen? That current will, the outer membrane will start to discharge. Now, this discharge current will flow through the resistor, but the capacitor will also start to depolarize. So, this voltage will be lost from here also. So, gradually, the voltage across capacitor will reduce, right? Eventually, capacitor will return to its neutral position and no current would flow at all through either resistor or capacitor, right? So the rate of discharging of this membrane, right? So once it has been charged, current has been moved outward. Now current was removed and discharging was done. So rate of discharging of this membrane is what we need to study in terms of time constant, right? So let's say the capacitor was charged to some voltage V naught, right? Now the capacitor is allowed to discharge. So the charge across capacitor is Q is equals to CV, right? Q is the charge, C is basically the capacitance, and V is the voltage. So let's say the charge changes by small amount. So whenever we are, this is pure physics now, right? So when something is changing by a very little amount, it is known as D uh, Q or delta Q, right? So from that delta only this D has come. So dq in time dt. So in a very small time, a very small amount of charge has actually changed. So it will also cause a small change in the voltage, which will be dv, right? So dq is equals to c dv, right? Because it happens in a very small amount of time, we can write it as dq by dt is equals to c dv by dt, right? Because we are adding dt across both left and right, it doesn't change or alter the equation. So the rate of change of charge is proportional to the rate of change of voltage, right? So that is what it means. Something per unit time. This is also per unit time. So rate of change of charge proportional to rate of change of voltage. Then we see how this discharging actually happens. So dq by dt is equals to c dv by dt, right? Because there is discharging, we will apply a negative sign, right? And over here we are replacing dq by dt by uh, current, that is negative v by r, right? So c dv by dt will be negative v by r because there is discharging. So the final actually terminology that we will get is dv by v is equals to minus dt by rc, right? Or we can also write it as v is equals to v naught E raised to power minus T by RC, right? And this RC is resistance into capacitance, which is also known as tau, right? So how this comes is, it because it is an exponential decay over here, T is equals to 0. So E, that is why E raised to power minus T by tau will also become 0. So V will become V naught. As t becomes larger, so the time becomes more, the fraction will become smaller than 1, so v will reduce, and t is equals to infinity, v will become 0, right? So this is tau. Tau is equals to rc, which is the resistance into capacitance. It determines how slow or fast the capacitor will charge. It is known as time constant for the membrane. So I understand that understanding tau without understanding the circuit of this circuit theme or in the neuron is a little difficult. But I promise that length constant understanding will be much easier than this, right? So time constant does not depend on the area of the membrane actually. It depends because if a larger area, this concept you can understand now, right? So it does not depend on the area of the membrane because if the area, if a large area is present, capacitance will be larger in proportion to the area, but resistance will be smaller, right? So capacitance and resistance are inversely proportional when it comes to area that we are talking about, right? So that is why it is not at all dependent on area, but it is dependent on properties of membrane such as density of ion channels, thickness of the membrane, etc. Right? So, T by tau is a dimensionless quantity. Tau has units of time since tau is equals to RC. In a neuronal membrane, resistance is in the order of mega ohms, that is 10 raised to power 6 ohm. That is a lot of resistance is there, and capacitance is in the order of nanofarads. That is very little capacitance is there. So 10 raised to power minus 9 farads. So tau will be in the order of milliseconds, right? When we calculate this. So, approximately, uh, at t is equals to tau, the the value of V will be 0.37, which means that V has become approximately 0.37% or 37% of its original value, or tau is the time in which V has reduced by 63%. So the rate of discharge is 63% when T is actually equal to tau, or this time constant. So if tau is larger, it will take more time for the membrane to discharge, while if it is smaller, it will take less time for the membrane to discharge. So tau indicates the amount of time it takes for a membrane to discharge, charge or discharge. Right, so that was a membrane time constant. This is the graph for discharging. This is the time graph for charging. It will just be the opposite concept of what we were looking at. So instead of that negative, it will just be positive. It's the only difference, right? Okay. I think length constant will be the final thing I discuss for today. I have more slides, but I'll probably include it in next week's lecture. Uh, yeah. So when we talk about conduction along the length of a neuron, right? It could be the length meaning how big the axon is, how long the conduction has to occur, right? That is what is going to determine the length constant. So, for instance, if you see, there are two major components when we speak about the length constant of a branch of neuron. It is either the membrane component or the axial component, right? So, membrane component is over here, what is around the membrane itself, and axial is, how far from the point of origin it is. So x is the distance. That is, if the current was applied in this region, then how far along are we actually taking the reading? If it is over here, it will be high. If it is over here, it will be lower than that. And lambda is the length constant, right? So the formula for length constant is delta v is equals to delta v naught e raised to 1 minus x by lambda, where x is the distance and lambda is the length constant. So how is it determined? It is also an exponential decay like time constant that we spoke about. The maximum value is reached when x is equals to 0. At the point of injection, that is denoted by delta V naught, right? The rate of decay is determined by this parameter lambda. So if lambda is equals to 1, it becomes value becomes delta V is equals to delta V naught e raised to power minus 1, or it becomes 37% of the maximum value, right? So if the lambda and the lambda is actually the length constant, it tells how much length does V reduce by six, in how much length V voltage reduces by 63%, right? So that is what it is indicating over here. So it depends on two parameters. First parameter is the membrane resistance, and the second is the axial resistance, right? So if the membrane resistance is more, uh, there will be less leakage and bigger lambda, right? So what it means is if the membrane resistance is more, meaning suppose we have a wire and it is very well insulated, right? If it is very well insulated, then it will have more resistance on the membrane because current will not be able to pass through it because there is insulation. So more resistance on that side. That is why the current will flow in a straight line rather than getting leaked from the ends or sides, right? So that is why higher membrane resistance will lead to longer trajectory of the current to flow, right? But if the axial resistance is more, right? That is, there is more hindrance along the length, then the length constant will be smaller, right? But if what you say is that it is flowing in this trajectory, but over here there is some hindrance that is present across along this trajectory also, then the length constant will be smaller because there will be a lot of resistance that will be coming, and whatever propagation of the current that has to occur will occur over a short distance because it is spending more energy during that region only because it has more resistance, right? Lambda is equals to under root Rm by Ra. So unit of lambda is meter actually. What is the relationship between length constant and axon diameter? This Rm depends inversely on surface area. Thus, Rm varies as 1 by 2 pi r. Uh, and depends inversely on the cross section area. Thus, Ra varies as 1 by pi r square, right? And Rm by Ra is proportional to the radius. In other words, length constant increases with radius. So, the thicker axons will allow conduction of electrical signals with less attenuation. So, the signal will be able to grow till a longer distance, right? So this is the time constant and length constant. Time constant basically defines how slowly the signal changes in a neuron, assuming a point or uniform neuron. And length constant basically states how far the signal spreads in a neuronal branch, assuming steady state, right? Then I had planned on discussing the insulation or the myelination that is present along these axons, but I think this part I'll be picking up in the next session. Um, Not right now. So for now, I'll just stop sharing the screen. If you have any questions or any suggestions pertaining to this particular time point, this particular session, you are free to let me know. So if there's anything, please do tell, or something that you want uh, to be included. Right, I'll just share the screen and I'll show you what else I had planned. As I told you, I had a lot of other things also planned, but not everything I was able to keep. So what I had kept was actually the insulation. Okay, okay, not much. Maybe I can just finish this part so that the things that I have anyway not been able to include over here as of now, that I'll be able to share next week, right? So probably I'll just go along with that. Few more slides. I think 10 minutes it should take. Yeah. So when we talk about an action potential, we also have to talk about these action potentials that are traveling very long distances, right? For instance, there are some that will originate from your brain and go probably till very far along, right? Also, so most most of these action potentials that are going towards the ends of your feet or hand are arising either in the CNS or in the spinal cord, and both of them are very far away from these limbs also, right? So how does this propagation happen? So that had something to do with our understanding of the time constant and the length constant also. Along with that, we should also understand how myelination actually helps us in in conduction across these axons, right? So when we talk of an unmyelinated axon, for instance, right, over here we see there is a cell body, axon, axonal end feet, dendrites in the cell body, and over here there is another part that is labeled over here, that is axon hillock, right? So why is this part labeled? Because axon hillock is generally the region where the action potential is actually initiated, right? So this is where impulse is initiated. So when we look at this region, this is what you can see. That why it is initiated over here? Because hillock has the highest density of voltage gated sodium channels, right? So the depolarization will be maximum over here, right? So it is site A over here is the site of original action potential, right? It is a region of depolarization. Then this basically transfer signal to site B, right? Site B will transfer it to site C. Why it will not go back to site A is because site A was in a depolarization state, right? After this depolarization state, for some time it will be in the refractory state. That is a state in which these channels cannot respond any longer, right? They have just come back to, they're just coming back to their original state, and they cannot respond as normal ion channels for some time, short period of time. In that short period of time, the signal will already have transferred from side B to side C, right? Assuring that this is a unidirectional phase that is occurring, right? Similarly, from site C, it will go to site D and not site B, because now site B will be in the refractory phase, right? And so the propagation will continue, right? This is what happens in the normal axon. Now, what is the problem over here? So, as it is going from site A to site B to site C to site D, what will happen? Some signal will keep getting lost because of them going forward also, and because it is open on the ends over here. So, that RM that we were talking about, some signal will be lost towards both ends also, right? Because there are channels present over here and here as well. So, what was the answer to this that was provided by nature itself was saltatory conduction, right? So, how this happens is if there is a myelinated axon that is present, right? So, these blue colors represent myelin over here, and you can see that in the duration of where myelin is present, it is essentially an insulation. So, RM over there is very low, right? Membrane resistance is very low. So what happens is that whatever signal is passing, it is not getting dissipated during in this region where this myelin coverage is there, but it is getting dissipated at this node of Ranvier region, wherein it feels like the signal has jumped from this node of Ranvier to this node of Ranvier. Why these nodes are also present is because these are open ion channels are present over here. It is also required for regeneration of that signal sometimes, right? So you can see that when we talk about when we look at it in a graphical format, what it looks like is that the time between stimulus and arrival of action potential, right? You can see that wherever these myelin myelin is present or the insulation is present, it is moving forward from this point to this point without much dissipation. But wherever there is a gap in this myelin, there is a lot of dissipation and not that much distance that is being transferred. Still, the dissipation is much more, and that keeps happening at each of these openings, right? So that is why it looks more like a saltatory conduction, that it is jumping from one point to the other, one node of Ranvier to the other node of Ranvier. So that is a summary or a brief description of how the signal is actually propagated. For now, that is all we'll be looking at. Yeah, I should stop sharing the screen also. Yeah. Uh, so if there are no questions for now, that will be all from my end. So in that case, I'll just stop the recording and I'll, uh,
Uh, Hello,
yeah,
ma'am.
yes, Diwakar. Yes,
Uh, so, first of all, thank you for this session. You spent your
yes.
energy. Felt like a special session because only five are available. And what are the frequencies that these sessions will be kept?
Okay, so I have actually, okay, so you will be getting a notification for two TA sessions actually. One is conducted by me. The series of sessions that I have kept it for every Monday from 7 to 9. And this is only week 3. So I have around 30 sessions that I have planned. So every Monday till then, from between 7 to 9, I'll be hosting these sessions, wherein what I'll be discussing is again the previous year questions, certain concepts related to that. And again, these were some things that were briefly touched upon during sir's lectures, but we can study about them in more detail over here, is what I thought. So, for instance, GHK equation, Nernst equation, also I thought I'll keep it over here. Um, but that does require more time. So, for now, I have not kept. So, those are the things that I have planned for these sessions at least.
Okay, so what are those others? Who will be taking the other sessions?
Yeah, so there is one other TA. I think he has planned his sessions on Saturdays, 3 to 5, if I'm not wrong. I am because we are not directly in touch. We are personally in touch with NPTEL actually for scheduling these sessions. So I am not aware of what exactly will be the course description for his session. But I think there will be recorded lectures also. Like for instance, I recorded this also because we need to provide that recording to NPTEL, who will also in turn provide it to you. So you can go through the previous recordings of both mine, his sessions. You will understand like what is the kind of syllabus that is being covered in each of these. So, but the timing I remember for the session, it is from 3 to 5 every Saturday. And
Three
there
to
will be
five.
a
Okay.
separate link for that actually. It will be available on your announcements page.
So these sessions are being recorded and
Yes, yes, yes. So we'll upload the recordings also. The recording link is also provided to you on the announcement page, if I'm not wrong.
okay, 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 ma'am. So I have one more question.
Yeah.
Uh, so my purpose of attending this course is understanding a conceptual flow. Uh, Now that we are studying more about history and how each neurotransmitters and about action potentials, about detail, and as someone
Yes.
who have amateur experience in this, so should I be practicing the drawing uh, for the exams? Like it looks like complicated, or it's going to just be the formulas or multiple choice basically.
will most likely be multiple choice, but we'll confirm that with the IIT Madras and we will let you know. Um, So drawings, I don't think you need to delve into. Conceptually, it should be more stronger that even if it will not be an MCQ, sir has kept a lot of MSQ questions, for instance, this time. So that will, in if you are conceptually clear with it, you will be able to answer those multiple select questions also, which are a little more trickier than MCQ, the traditional MCQ questions. So that will probably be how your
So more
final
than one of the
exams
options
will
are
also
going
be.
to be right.
Yeah, yes,
So yeah.
yes.
So I've already attended those in assignment, and
Yeah,
for which they are also graded based on how much we are
how
selecting.
accurate you are. Yeah,
Yeah. Yeah. If I'm not
yes,
selecting the one option, they are reducing respective marks for that. Like,
yes. Yeah. So at least it is respective marking. I did not know that because in most of the competitive exams and all, you do not get a mark if you don't get all of the correct once, right? So even if it is that way, it is good for you only if that is the marking system. But yes, they are a little more tricky. You do not need to delve into drawings and all. Conceptually, if you understand, it will be enough.
yeah, cool. There is one more question which is last.
Yeah.
So I'm also thinking about doing masters in Europe. So,
Okay.
which is the reason I'm doing this. So, I'm also thinking about applying for research assistant or intern in some other
Mm.
facility which is like within India. So, would you suggest something which aligns with neuroscience, cognitive psychology? So, which is open for amateurs. So, I don't have a prior certification or nothing. So, I'm basically from
That is
engineering background. So,
okay. That is completely all right. Even if you do not have prior experience to it, what you can always do is, yeah, you do need to sit for exams and all if you want to apply for an MTech. But for training positions, you can always apply. You can apply for internships and training positions. For that, what you can do is, you can always write to people who have labs based in this neuro setup, right? For instance, like how I would suggest you to go about that. Where do you live, for instance?
so I'm from Chennai.
Okay, so Chennai is like one of the metropolitan cities, right? There are so many institutes in Chennai. So, what I would suggest is first look within your city, right? Because it is more of an internship or training position. You gain experience, then you go for some JRF position wherein you actually get paid. So there for that kind of a position, you look at institutes within Chennai, IIT, for instance, or this. Oh, that is Bangalore. NCBS is in Bangalore. I was going to say NCBS. Okay. So other institutes in Chennai also you can look at or in close vicinity to your place. And what you can look at is just go through the electrical department. The What is your background in engineering also?
So it was from automobile. So I have experience in
Automobile.
race field.
Okay.
So as a motorsport engineer.
Okay, okay. All right. I thought if it was electrical or mechanical, probably you could do an interdisciplinary thing. Like you can focus on labs that are interdisciplinary. Okay. So you can actually focus on, you can try to search for instance, you are going to IIT Madras website and you look for the biotech, biomed, bioengineering, the chemical sciences, and often electrical sciences also. These departments you look at. I don't know if they have a cognitive science department. We do at IIT Kanpur. If they have one, then over there also. So these are the departments you need to look at. Once you open the department web page, you will see the people section or the professor section or the lab section, right? Wherein you can it is like 30 minutes you need to spend on one particular department, right? You will just be able to filter out whether a particular lab is working on something related to neuro or not, right? Just jot down those labs that are working on it and just send uh, 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 a mail to all those supervisors, right? With your CV, with your motivation letter, you just send a CV to all supervisors. If at all they have an opening in your lab or a possibility for a trainee in their lab, they will definitely respond back to you. Uh, and if you're very interested in a particular lab, you can always try to reach out to members of the lab through LinkedIn to understand how better to, obtain that position or if that position is available. So that is like how to go about it.
Yeah, ma'am, I got a good idea. Thank you so much.
Yes. Okay. So if that is all, we'll end this particular session and yeah.
Yeah, thank you, ma'am. See you on next Monday.
Yes. Sure.